Now I will introduce our first speaker, which is Silvia Scaglione, and Silvia received her PhD in bioengineering from University of Genova in 2005, following a year as a visiting scientist at the University Hospital of Basel. Since 2010, she has been a permanent researcher at the National Council of Research in Italy, becoming a senior researcher in 2021 and leading the tissue engineering team. She is the author of more than 80 international peer-reviewed papers, book chapters, and holds 7 patents. She is a member of international scientific committees such as the International Microphysiological Systems Society. In 2017, she founded the React for Life, an Italian biotech company that develops organ ownership technologies to accelerate translational research and improve the reliability of drug testing. React for Life has won several international projects and awards, including the EU Innovation Radar Award of Best Health Technology and the CPHI Award in 2023. She is also an ambassador for the European Innovation Council, and today she is going to talk about MPS and MIVO, multi-in vivo organ ownership, advancing translational research and drug testing. Silvia, thank you very much for having accepted our invitation, and the stage is yours.
Thanks for your kind invitation and introduction. I will share the screen and we can start. Super, thanks a lot. Yeah, I will speak about microphysiological system, and in particular about the MIVO multi-vitro organ on a chip platform that we have been developed and used for translational research and drug testing. About myself, you have already nicely presented my story from the PhD in bioengineering, and then when I received the permanent position in National Research Council about 15 years ago, leading the group of tissue engineering, and then more recently, about 9 years ago, I founded React for Life. Currently, I'm chief scientist and president of this company, where we develop a new organoid chip platform, and we do high-level research in different fields of organ on a chip. Basically, this story started from the visualization of an important need, meaning the request to develop humanized experimental models to recreate the real human biology or physiopathology in vitro. Typically, the current available models are either 2D simplistic models or animals, but in both cases, we still have some issues in properly recapitulate complex diseases, in resembling cell-cells interaction, in particular immunocells interaction with the tissues, properly resemble drug diffusion into the tissues, finally affecting toxicity and efficacy readout. And for all this reason, scientists and pharmacologists still are looking for better models. They have tried to use three-dimensional tissue models like scaffolds, bioprinting, more recently organoids, but still there is a missing pieces, which is the fluid flow, the presence of the fluid flow, which properly feed 3D tissue models, where we can introduce circulating cells like immunocells, but also compounds, soluble compounds, drugs, to properly recapitulate the realistic exposure to drugs of our human tissues in our body, and also to properly connect different organs towards a more concept of body on chip. And therefore, the missing piece of this story is really the microphysiological system. In particular, in React for Life, we have patented MIVO, which is the acronym of multi-in vitro organ. Basically, it is a platform which enables to host these 3D tissue model that I mentioned before with a millifluidic flow conditioning driven by a pumping system. And in this flow, we can have circulating cells, we can have drugs, and eventually we can also fluidically connect multiple organs, as you see in the cartoon. This approach enables to keep the benefit of the humanized cellular models, but introducing a fluid flow which is more systemic and more predictive of what happens in clinical stage. We have validated this approach and this platform in different fields, in oncology, but also in the field of gut models for pharmacokinetics purposes, but also for complex disease models. We have been optimized many different barrier tissue models and additional validated organ models. But today I would prefer to focus the attention on oncology, where we have a strong experience since many years working with many different types of cancer models, from neuroblastoma up to the colon cancer model. Basically, one of the most important validation that we decided to fix as a starting point was a cross comparison with the animal model, which still represent an important gold standard, especially in the drug efficacy field. Therefore, we decided to use an ovarian cancer model in vitro in our MIVO organoid chip platform, but also in the static in vitro model, meaning growing ovarian cancer cells in a 3D matrix and placed in a well instead of within the organoid chip. And the same cells were injected in mice model. We waited the growth of the tumor, and then we administered the drug, cisplatin chemotherapeutic agent, in all these three conditions. Interestingly, we have seen that while in steady condition, after the administration of the drug, we observed still many proliferating cancer cells staining in green within the tumor, and very few dead cells in external area of the tumor stained in red. The same tumor model treated with the drug in the organ on a chip displayed a very nice distribution of dead cells within the matrix. And this was due to the completely different diffusion of the drug. So in other words, when the drug was placed in circulation in the organ on a chip, the presence of the flow resembling the bloodstream was able to properly diffuse the drug into the tumor, and therefore we induced a reliable and predictive tumor cell apoptosis. And this is the reason of why we obtain a very similar regression of the tumor in the MIVO organ on a chip and in animal model, the black bars, but not using the standard simplistic static model. So the take-home message of this slide is that, especially when you work with a three-dimensional tissue model, the presence of the flow becomes mandatory in order to properly diffuse the nutrients, but also to properly diffuse the drug. Otherwise, the readouts can be very different than the real clinical data. More recently, one of our customers in Thailand has used a similar approach. So he wanted to test the efficacy of different compounds against another type of cancer model. It was an oral cancer model. But the beauty of their paper is that they combine a three-dimensional bioprinted tissue resembling the oral cancer model with the coexistence of bacteria. of oral bacteria in order to resemble an oral mucositis disease conditioning that they observed in patients during the chemotherapy treatment. So they first nicely recapitulate this complex disease with a coexistence of the tumor and the bacteria, and they appreciated in dynamic condition using the MIVO the upregulation of specific markers of this tissue. And then they used the organoid platform for placing in circulation different drugs using also different doses, and they assessed the efficacy of this drug to reduce the viability of tumor cells, and nicely they observed a dose-dependent regression of the tumor. The capacity of our platform to fluidically connect multiple organs, it's very interesting when the focus of scientists is related to the progression of the tumor towards metastasis. So in many cases, the focus from the scientific, but also from a pharmacological perspective, is not necessary to reduce the primary tumor that typically can be surgically removed, rather than reduce the capacity of a tumor cell to escape by the primary tumor and become circulating tumor cells, finally reaching a secondary tissue model. The platform enabled to properly set up the velocity and the shear stress induced by the velocity, and therefore you can really monitor also the role of circulating tumor cells in remaining viable at different flow conditions. Moreover, more recently, the University of Turin published a paper using our platform. They were interested to study and to assess the efficacy of different treatments, so standard treatments and immunotherapy treatments against the pancreatic aggressive tumor model. They appreciated the possibility to have multi-organ connection in order to assess and quantify the number of tumor cells clones escaped by the primary tumor in the tumor chamber and separately in circulation. So here you see results in the tumor chamber, while here we have the results in circulation. And the bars show the number of new cells, tumor cells clones after the treatment, so the purple and the green bars, and even before the treatment as negative control. So it is a very interesting and quantitative readout enabled to assess and quantify the efficacy of new therapies in reducing the capacity of some cells to escape by the primary tumor, become circulating tumor cells, and aggressive towards a secondary tissue target. The multi-organ connection becomes also very interesting if the focus is to simultaneously analyze the efficacy, the desired efficacy of the drug against the tumor, but also the potential off-target toxicity. So in this case, we co-cultured an ovarian tumor model fluidically connected to the healthy liver model. We placed in circulation the drug, and we simultaneously analyzed at different doses the capacity of the drug to induce a cytotoxicity against the cancer as desired, but also monitoring potential hepatotoxicity known for this drug. And nicely, these results show that when we had these organs fluidically connected, there is a sort of a crosstalk and cross-protection among the organs. And at the end, the results under these conditions are much more closer to the in vivo results compared to the efficacy or toxicity assays measured using a single organ configuration. This is extremely important because you can also fluidically connect multiple organs, more than two. But one of the most important and fascinating validation that we have carried out using the MIVO organoid chip is what is related to the immuno-oncology. So the platform, as you can see in the picture and in the cartoon, enabled to host a three-dimensional clinically relevant site tissue model, while we have immunocells in circulation. Typically, the tumor model are placed in an insert, which is placed into the chamber. The insert has a permeable porous membrane, and we can place at the beginning immunocells only in the basal compartment in circulation, but we have the capacity to collect the media around the tumor and quantify the number of the immunocells that are able to leave the flow, win the gravity, and going up. And interestingly, this percentage is close to clinical data. In this case, I'm speaking about a neuroblastoma pediatric tumor model and natural killer cells in circulation. This work has been carried out together with Gaslini Hospital in Genova. So they have also clinical data available for cross comparison. But nicely, when we didn't place any tumor model in the cancer on chip, meaning the negative control, we didn't find any immunocells above. So in other words, immunocells flow in circulation, physically separated by the tumor compartment, and they decided to infiltrate the tumor compartment only when they receive the signaling by the tumor. And when we didn't have any tumor, they do not receive any signaling, and they continue to circulate. And at the end of the experiment, we could collect the tumor out. We did some confocal imaging, finding immunocells even within the tumor. And then we did some cytofluorimetric analysis to assess and quantify also the amount of tumor cells in apoptosis induced by the activity of the immunocells, and also to monitor immunocell phenotype. A similar approach has been also used by the University of Florence, that they used our platform for co-culturing breast cancer cells with lymphocyte T cells primed by the maturation of the dendritic cells that were treated with a bioactive immunomodulating agent. So this is something towards the possibility to test immunomodulating agents. So we find that these bioactive compounds are internalized by dendritic cells. They primed T cells, and then when we place the T cells in circulation, again we found an increasing of T cells infiltration in the tumor and an increasing of induction of apoptosis as effect of these immunomodulating agents, and also as effect of the adoption of this reliable organoid chip platform, which supported the co-culture of 3D tumor model and circulating immunocells. A more recently work in this field has been carried out by a hospital where they were interested to study a very new approach, a cell therapy approach coupled with boron neutron therapy. So the idea was to target the tumor and then to deliver cytotoxic compounds. This was very fascinating, this paper, because they used tumor-infiltrated lymphocytes as a Trojan horse to deliver into the tumor this boron-containing compound, and after that they were able to irradiate the tumor with a beam of neutron to generate a very effective destroyment of the tumor itself. And they appreciate the possibility to use the cancer on a chip for demonstrating the infiltration of the tumor-infiltrating lymphocytes into the tumor. We are also proud to see that not only academic research, but also pharma companies are now ready and prompt to adopt this technology for testing their drugs during the preclinical stage. This is also what Roche has recently done. They have tested one of their immunotherapeutic agents using our platform. But the beauty of this approach is that they appreciated the possibility to culture ex vivo pieces of tumor biopsies. So they did an extensive patient stratification and through RNA-seq readout, and they were also able to discriminate among responder and not responder phenotype. We have, of course, other models, as I mentioned before. So these last slides are just to give you some flash regarding other potential application of microphysiological system. In the gut, it is very important to have the possibility to test the interaction between the gut and microbiota, as we have already done, and also to introduce immunocells, also for inflammatory studies. And this is appreciated not only for PK studies, but also for safety and for efficacy investigations. Also the skin, it's an interesting barrier tissue that can be hosted in our skin on chip, and also in this case we could use this approach for permeation, but also for safety context of use. In this case, of course, the culture is a higher liquid interface culture, but the platform enables also to manage this type of configuration. So in conclusion, we can say that this organoid chip enabled to combine the possibility to host three-dimensional tissue models like biopsies, keeping the real heterogeneity of the tissue for more predictive results. We can place immunocells in circulation, investigating the all immunorelated disease, but also testing the all immunomodulating agents. We can fluidically connect multiple organs for systemic disease modeling, for more complex bioavailability assays, and for investigation of metastasis paradigm. And of course, we are fully aligned with the 3R approach and to the approaching regulatory changing, which foster the adoption of this approach. The last slide is just to comment that from the practical perspective, the most common feedback of our end users is that the MIVO, it's really simple to be used, and it enabled to keep the cellular models already established in the lab, just move on in a fluid dynamic configuration. So there is no need to start from the scratch, but also for those that are using organoids, complex scaffold-based models, you can very easily add the flow and go on. And I want to thank all the people involved in these validations and the European Commission for the grant that support also our research.
Thank you so much, Silvia. It was a very nice presentation. Does anyone have a question for Silvia?
Hi, Silvia. Very nice talk.
Thanks.
Very, very interesting. So, unfortunately, I work in a different field. I work in retina diseases. And my question is if you have kind of any project, or did you I think this kind of platform can be used for our questions as well. But my question is if you have explored something related with retinal organoids, retinal as chip models.
You are lucky because as a company, we typically do monthly some webinars for our community. And the last webinar was exactly on retina on chip. So we can also share this the recording of the webinar. So it was,
sorry? So great.
I can send you this link, and there is also on our website. There is a professor of the University of Buffalo in United States. She's studying retinal tumor models, and she appreciated the possibility to culture these cells under fluid dynamic condition. She explained the importance of the flow to properly recapitulate this complex niche. And she is obtaining very interesting results. This work is within a grant she has obtained in United States. And there is also this type of application.
Thank you. Thank you. I will see the webinar. It's very interesting for us as well. Thank you.
Thank you, Sandra. Maria?
Hi, Silvia. Thank you for the presentation. So I have two questions. One regards to asking if you have been using these models to study infection by bacteria.
Yes.
And how do you do it in these multi-organ?
Yes, we have used internally bacteria, and we have an European grant where we are using virus as well. Depending on the type of the tissue, the virus or bacteria are placed apically or typically apically, and internally bacteria have been applied in coexistence with the gut to mimic gut dysbiosis or to the vaginal tissue. But similarly, it can be applied also in the lung or other tissues. And typically, we assess this disease in terms of damage of the barrier function, eventually passage of the barrier across the tissue, so we can collect in the basal compartment if we find bacteria even there. This is something that it might happen also in IBD, inflammatory bowel disease models. And so, we have also a line in this direction.
Okay. And so it is also possible to do like efficacy studies, right?
Yes.
With drugs, I mean.
Yes, we have recently submitted, not yet published, a paper where we fluidically connect the gut with a dysbiosis connected to the cancer. And we assess the drug efficacy, so a chemotherapy in circulation, and we checked the role of the dysbiosis in the gut in reducing the efficacy of anti-cancer drug in the cancer. So we have seen that there is this reduction of efficacy, and this is mainly related to the induction of inflammation by the dysbiosis, which becomes a systemic inflammation. So it is moved from the local gut niche to a systemic inflammation. So at the end, we have a changing of the tumor cells behavior that become more resistant to the chemotherapy.
Okay. So thank you. And so regarding, I'm particularly interested in your skin ownership model. So what kind of model it is? It is epidermis model, or it is a full skin model? And
depends.
What type, you have both.
It really depends on the question. Typically, we prefer to use 3D tissue models with epidermis, dermis, and sometimes also hypodermis if the focus is also transdermal delivery of vaccine or whatever. We have worked with biopsies from human or from animals or reconstructed tissues.
Okay, thank you.
Thanks for the question.
Thank you. Duarte?
Hi, nice talk. Thank you very much. So regarding the metastasis model with the CTCs, so can you also have a barrier that simulates the endothelial barrier to simulate intravasation and extravasation?
Yes, we typically place endothelial cells in the lower part of the insert because we have seen that the presence of the flow enable to accelerate their maturation, differentiation, or organization. So at the end, they have a higher vimentin expression, and then we have also already placed circulating tumor cells, and we have seen their capacity to pass across the endothelium, mimicking the moment when tumor cells decided to extravasate. Yeah.
Thank you.
Thank you. Sarella?
Hi, very nice presentation. Thank you very much. I'm more interested in the gut, so we have organs for everybody. I was wondering how long do you manage to co-culture the bacteria in contact with the epithelial cells?
It depends. So when we work with cell lines, we are able also to keep them for weeks. We have also seen that when we use reconstructed gut tissues, that typically it's suggested to use within the first days, otherwise they lose the barrier function. The presence of the flow enables to properly feed the tissue, remove extra mucus, and we are able to keep in culture for 3 weeks. Without losing the barrier function. So it's very common to observe a strong increasing of the longevity of the tissue to the presence of the flow.
But the flow is in both sides of the...
In the gut, we use in the both sides. You are right. In the gut, since we want also to recapitulate the gut lumen peristalsis, we have the flow even in the apical side. And this is important to remove the extra mucus, but also if you have bacteria, to keep bacteria in a nice coexistence with tissue without a suffering of the host tissue by the strains.
And then just last question, in the top, when you have the flow, you can have that flow anaerobic medium, or is
This is a good question. The platform currently is not sealed, so if you use an anaerobic, an epoxic cell culture incubator, the epoxy condition is transferred into the chamber. We are also working on trying to managing a gradient of oxygen from the apical to the basal within the same incubator.
Yeah, okay, so last question now. So when you have I guess you tested this model with CACO2 cells? Yes.
Not only CACO2 and HT29, CACO2, HT29 and fibroblasts, depends.
And then how long do they take to differentiate? Because in the static...
they take 3 weeks. More or less. In dynamic, already after 8-10 days, we have tight junction formation, so one expression and barrier function on the peak.
Okay. Thank you very much.
Thanks, Sarilla.